Good evening, my dear aspirants. We are again back on your own platform of Biology Nagari with a new concept of spermatology. Basically, we are going to study spermatogenesis. What is basically the spermatogenesis? Spermatogenesis is basically the process in which the sperms are being prepared inside the testis of a male. Or we can also say that the formation of a sperm inside the seminiferous tubule of the testis of a male. Because basically when we talk about the gametogenesis, there are spermatogenesis as well as oogenesis. In the case of females, the formation of female ova inside the female's body is considered as oogenesis, which is basically the gametogenesis of female. But in this lecture we are going to talk basically only about spermatogenesis. It means the gametogenesis in males, that how the sperm are being prepared inside the seminiferous tubules of the testis of a male. So basically when we start talking about spermatogenesis, this word sperm, basically sperm, yes, the male sex cell. Genesis means formation. Basically the formation of sperm is called spermatogenesis, and where it is being happened, spermatogenesis is being occurred in the seminiferous tubule of testis. And we know that we have already studied that testis is found in the extra abdominal pouch, which is the scrotum. These are two in number, paired. Basically these are paired, found inside the scrotum, and inside this testis there are basically 2 to 3 seminiferous tubule per compartment in per testis, and there are basically huge number of compartments inside per testis, and in that, among that compartments, 2 to 3 seminiferous in each compartments of each testis. Now see what happens basically. Basically when we see about the seminiferous tubule, where it is being going to happen, this is basically the beta seminiferous tubule. Inside the seminiferous tubule there are basically two types of cells. First is called the germ cell, which is basically called spermatogonia. Second one is the Sertoli cell. So there are two types of cell found inside the seminiferous tubule. First one is the germ cell, which is basically the spermatogonia. Second one is the Sertoli cell. Basically the Sertoli cell provides nutrition to the germ cell. Nourishment to the germ cell. And there are some cells outside, at the outside region of the seminiferous tubules, which are called interstitial cells or Leydig cells. These are called Leydig cells. Basically these Leydig cells releases male androgen, which basically stimulates or starts the spermatogenesis. Now see how these all spermatogenesis is going to start inside the male testis. Note it down. See beta, basically this process of spermatogenesis inside the testis of a male is going to start at the age when the age hits the puberty in male. What is puberty? Yes, puberty is the onset of sexual maturity. When the age of a male hits the puberty, which is basically 13, 14, 15, this is the age range. Basically what happens? The hypothalamus, yes, I am talking about the master endocrine gland, the hypothalamus, starts releasing GnRH, which is basically the gonadotropic releasing hormone. This gonadotropic releasing hormone goes and acts on the pituitary, basically the anterior pituitary, and by the act of GnRH on the anterior pituitary, it starts releasing gonadotropins, which are basically LH and FSH. LH is a luteinizing hormone, FSH is a follicle stimulating hormone. It means, I am repeating my sentence, The hypothalamus releases gonadotropic releasing hormone, which is an hypothalamic hormone. This hypothalamic hormone, gonadotropic releasing hormone, acts on the pituitary, basically the anterior pituitary, which is called adenohypophysis. This adenohypophysis start releasing the gonadotropins, which are the LH and FSH. These are the two gonadotropins. These FSH and LH basically initiates the process of spermatogenesis. So this was how the spermatogenesis was initiated in the male when the male hits the age of puberty. यही बताना चाहता था बेटा कैसे मेल में spermatogenesis puberty एज पे आते ही स्टार्ट होता है. What was the role of hormone here? अब बेटा आगे देखेंगे. Basically what happens? जब I am telling that the process of spermatogenesis is going to happen inside the seminiferous tubule. So let's see a view on seminiferous tubule. Better if it is a view of seminiferous tubule, the inner lining contains the germ cell and also the large sized cells, which are the Sertoli cells. These are the germ cells and these are the Sertoli cells. These are the Sertoli cells and these are the germ cells. And we know that this Sertoli cell provides nutrition to the germ cell. Now see what happens. These germ cells, which are basically called spermatogonia, which is basically the sperm mother cell, or we can say the primordial germ cell. These spermatogonia are basically where the spermatogenesis is being started. It means this spermatogonia, I am writing here, this spermatogonia, which is a male germ cell actually, or we can say immature sperm, okay, goes through the mitotic division, and increase in number. There will be increase in number. Some of these spermatogonia converts after the differentiation into primary spermatocyte. These were the spermatogonia, and some of the spermatogonia after differentiation reaches the stage of primary spermatocyte. Further, this primary spermatocyte goes through meiosis first, and we know that the meiosis division completes in two stages: meiosis first and meiosis second. Here the meiosis first is going to happen, and we know that meiosis first is basically the reductional division. It means there will be reduction in the ploidy level. What happened when we talked from earlier? The ploidy level at the stage of spermatogonia was 2n. It means there was 46 pair chromosomes. Ploidy level at the case of प्राइमरी स्पर्मेटोसाइट, चूंकि यहाँ पे माइटोसिस हो रहा है बेटा, तभी तक रिडक्शन नहीं होगा, ये बना रहेगा, मेंटेन है, क्योंकि माइटोसिस क्या है, इक्वेशनल डिवीजन है, यहाँ पर मेंटेन रहने वाला है. तो यहाँ पर प्लॉइडी लेवल 2n मतलब 46. अब देखना आगे. This primary spermatocyte goes through meiosis first, which is reductional division, so there will be a reduction in the ploidy level, but hence increase in number of cells. डिवीजन है तो नंबर ऑफ सेल्स तो बढ़ेंगे, लेकिन प्लॉइडी लेवल यहाँ घट गया. क्या हो गया? घट गया. अब these are called the secondary spermatocytes. secondary spermatocyte कह दिया मैंने. अब देखना बेटा, ploidy level has been decreased and reaches to n. n means 23. Yes, 23 pair chromosomes. Now each of these secondary spermatocyte increase in number through meiosis second. Here will be a meiosis second, and we know that meiosis second is very much similar to the mitosis. It means there will be maintenance of the number of chromosomes. Ploidy will be maintained. So here n, these will also n, and these are further called spermatid. These are spermatid, but still now these spermatid has not achieved the proper shape of spermatozoa. So after differentiation they will achieve the shape of spermatozoa, or we can say the sperm, and further these are after differentiation called sperm or spermatozoa. spermatozoa, and this is finally the matured sperm. So what type of divisions we have seen in this process of spermatogenesis? This was the mental. Okay, and now further our sperm are formed, and this was the spermatogenesis. मुझे लग रहा है ये डिवीजन क्लियर है आपको, ठीक है? इसको अच्छे से देख लीजिए एक बार। अब देखिए बेटा, एक एक चीज हमने स्पर्मेटोजेनेसिस का अकॉर्डिंग टू एनसीईआरटी बुक मैंने इसलिए आपके साथ रखा है, और ये इंटरफेस आपको मैं यहाँ पर खोल भी दिया हूँ, देख लीजिए, ठीक है? आप यहाँ पर इंटरफेस देखते रहिएगा, ठीक है? और मैं आपको एनसीईआरटी में दिखा देता हूँ। बेसिकली एनसीईआरटी की एक लाइन इंटरफेस पे ध्यान रखिएगा। एनसीईआरटी की एक लाइन हमने क्लियर कवर किया कि इस पे दिखाते चलता हूँ मैं आपको। बेसिकली the production of sperm by spermatogenesis. अब एक और चीज आपको बता देना चाहता हूँ, the spermatogonia which is basically present on the inner wall of seminiferous tubule, inner wall of seminiferous tubule पर है ये germ cell spermatogonia, multiplies by mitotic division. इनमें mitotic division हुआ जिसको हमने equational division भी कहा, and increase in number, इनके number increase कर रहे हैं। इनमें से some of the spermatogonia changes into primary spermatocyte, इनमें से कुछ primary spermatocyte जिसका Ploidy level contains 46 chromosomes. Some of the spermatogonia primary spermatocyte में बदला। Further ये primary spermatocyte में first meiotic division, देखिए बेटा meiosis first मैंने कराया है, which is called reduction division. First meiotic division, reduction division leading to the formation of two equal haploid cells. दो equal haploid cells बना दिया, n मतलब haploid। तो देखो बेटा इसी को secondary spermatocyte कहा हमने। अब फर्दर दिस सेकेंडरी स्पर्मेटोसाइट, इसमें क्या हुआ? मियोसिस सेकंड, जिसमें बॉडी लेवल मेंटेन रही, तो 4 स्पर्मेटिड बना, लेकिन अभी तक इसने शेप नहीं लिया था स्पर्म का, तो स्पर्म का शेप लिया तब जाकर के स्पर्म का, तब तक स्पर्मेटिड ही कहा, स्पर्म को स्पर्मेटोजोआ भी कहते हैं। अब देखो बेटा, यहाँ पर एक चीज और बता देना चाहता हूँ मैं, ये जो प्रोसेस था स्पर्मेटोगोनिया से स्पर्मेटिड तक का, इसको साइटोजेनेसिस बोलते हैं, और ये स्पर्मेटिड से स्पर्म में डिफरेंशिएट हो जाना, इसको स्पर्मियोजेनेसिस कहते हैं। और इन दोनों प्रोसेस को साइटोजेनेसिस और स्पर्मियोजेनेसिस, इन दोनों को मिला करके कंबाइंडली स्पर्मेटोजेनेसिस कहते हैं। इज इट क्लियर? मैंने इसलिए बताया क्योंकि दोनों चीजें दी हुई है। मतलब स्पर्मेटोगोनिया का डिवीजन टिल स्पर्मेटिड, इट वाज कॉल्ड स्पर्मेटोसाइटोजेनेसिस, फिर साइटोप्लाज्म डिवीजन होता चला गया, उसके बाद ये स्पर्मेटिड डिफरेंशिएट हुआ है, इस तरह का डिवीजन नहीं हुआ, सिर्फ डिफरेंशिएशन हो रहा है, अपने साइज को अचीव कर रहा है, प्रॉपर शेप को, फॉर द लोकोमोशन टेल्स बना रहा है, इसको स्पर्मियोजेनेसिस, दोनों मिलाकर स्पर्मेटोजेनेसिस। अब सब स्पर्म में पूरी तरीके से बन गया, तो अब इसको रिलीज कर देंगे, व्हिच इज कॉल्ड स्पर्मिएशन। इस स्पर्मिएशन पे आपके एनसीईआरटी में दिया हुआ है, ठीक है? तो बेसिकली मैंने आपको दिखा दिया कि इन सारे के सारे लाइन को क्लियर कर दिया, और नीचे जो दिया है जीएनआरएच एक्ट करता है पिट्यूटरी पे, पिट्यूटरी से एलएचएफएसएच निकलता है, और ये एफएसएच लेडिक सेल्स पर एक्ट करेगा, और वहां से स्पर्मेटोजेनेसिस स्टार्ट करेगा। सारा मैंने आपको पहले ही बता दिया, ठीक है? अब देखो बेटा एक चीज, ये जर्म सेल से बन गया क्या? स्पर्म, ठीक है? लेकिन तो भी स्पर्म अभी रिलीज नहीं हुआ पूरी तरीके से मैच्योर, तो बनते ही ये प्रोसेस होते ही ये स्पर्म का हेड इस सर्टोरी सेल में इम्बेड कर जाएगा। देखो बेटा कुछ इस तरीके से, क्यों? क्योंकि ये सर्टोरी सेल इसको न्यूट्रिशन देगा। जब सर्टोरी सेल पूरी तरीके से न्यूट्रिशन पा गया स्पर्म, तब जाके स्पर्म को रिलीज किया जाता है, एंड दैट इज कॉल्ड स्पर्मिएशन। पूरी तरीके से जब हो गया तब इसको निकालेंगे, स्पर्मिएशन। ये दिस वाज द कांसेप्ट ऑफ स्पर्मेटोजेनेसिस। आई होप दिस इज क्लियर। क्लियर बेटा?